So first let's have a look at just what RNA is. So RNA stands for ribonucleic acid because it belongs to the nucleic acid category in biological molecules. RNA is a single-stranded polymer molecule, and polymer means large molecule made of many repeating units. And this single-stranded polymer molecule is made of RNA nucleotide monomers. So the monomers, the small repeating units that make up the large polymer, are the RNA nucleotides. So RNA molecules are shorter than the DNA molecules. And there are three types of RNA, and these include messenger RNA, ribosomal RNA, and transfer RNA. But you'll be learning more about what they are and what they do later on. So RNA nucleotides. The RNA nucleotides, on their pentose ribose sugar, they have a hydroxyl group on their carbon too, unlike the DNA nucleotides. And this means that the RNA nucleotides are not deoxy like the DNA. One of the main differences of RNA nucleotides to DNA nucleotides is that they have a uracil base instead of a thymine. So the uracil would be complementary to the adenine instead of the thymine. A similarity is that they still form phosphodiester bonds between adjacent nucleotides. So one of the questions is, why do RNA nucleotides and RNA molecules use uracil instead of thymine? And one of the reasons is that uracil requires much less energy to make than the thymine base. And as we produce much more RNA molecules than DNA molecules in our lifetime, by producing uracil, we save much more energy. Also, uracil doesn't maintain as much integrity as the thymine, so the information isn't as trustworthy per se. But this is all right for RNA's purpose because it doesn't need to maintain such high fidelity information as the DNA does. So why don't we use uracil in DNA? And this is because cytosine degrades into uracil. So if the cytosine degrades into uracil, it means that the DNA polymerase, when it is proofreading, if there is a mistake where cytosine has degraded into uracil, it would be harder to pick out the mistakes if uracil was a base in DNA as well. It wouldn't know which base to change back. So if you see here, this cytosine has degraded into uracil. If DNA polymerase came along and saw that there was a mutation in this base pairing, if uracil was a base pair in DNA naturally, it wouldn't know which one, the uracil or the guanine, which one had been a mutation, so it wouldn't know which base to change back. So this means that the integrity of the coded information in our DNA wouldn't be as strong as if we had thymine. So as I said, uracil mutates much easier than the thymine, so it has a lower integrity of information. And thymine maintains this integrity of coded information, so that is why it is used in DNA.